I am very grateful to the organizers and to anyone listening to this talk for being able to take part in a seminar that is directed at least partly to my work. So thank you. But it's also a great opportunity to honor my former brilliant student, Nongjain Tao. And I want to talk about the way in which Tao's research and mine have interacted over nearly the past 40 years. Now, we are honoring Nongjian in view of his untimely death, but I just want to mention here that I have lost some brilliant, remarkable colleagues and great friends at ASU, shown here, be familiar to many of you. I want to make the point that ASU has been a wonderful place to have a scientific career with great faculty colleagues like these, fantastic students and postdocs, of whom you'll hear a bit. I'll mention those that impinge on my interactions with Nongjian, but you'll see pictures of many others, and I could tell you all sorts of stories about all of them. But let's focus on Nongjian. So I came to ASU in 1979, and so here some years later is the group on its way to the APS meeting, a March meeting in Las Vegas. And you see here Nongjian, if you can believe it, this is a young Nongjian in 1986, not long after he had joined the group. And like the rest of the group, he was working on laser spectroscopy, in Nongjian's case of DNA and the hydration shells, and he graduated with a thesis on that subject in 1988. I've pointed out one other group member here because he is the exception to the laser spectroscopy rule, which is Ben Barris, who just joined me as a master's student and was helping me build the first STM in my lab. So this marked our move into nanoscience. So here is that STM, and let me just play little snippets here of a movie made by Mervyn Miles when he visited my lab in 1986.
You see that central micrometer there is mounted on a sort of cruciform of 1, 2, 3, 4 ceramic piezoelectric elements, which scan the central part. And then there's a fifth one down below for the Z motion. Then finally, the samples advance towards the stage. There's a 20-to-1 lever in here that rocks the sample up like this, and that's the piezoelectric motor. That's all there is to it.
And what about your vibration?
Well, this is one stage of vibration isolation, and this table is an air table that's a second. That's it.
Yeah, so we'll race ahead of that a little bit. Okay, so here's the tip viewed through a microscope. Here are the electronics taking up a whole rack. I'm lowering the piezo and we're getting the microscope into tunneling. And so we're ready to start recording some traces. And quite remarkably, as soon as this microscope got into tunneling, you could see atomic corrugations. Hard pressed to do that with a commercial instrument these days. This was the genesis, of course, of molecular imaging, which was formed in 1993. And I know a number of people in the electrochemical community have used MI microscopes. These are some of the great colleagues I had there, in particular, Tim-Wei Jing, and this is Peter Hintendorfer for the University of Linz, where we're being honored for technology development.
You see
So let me turn now to the reason we built the microscope, which was to use in biophysics in the hope of imaging biological molecules in water. And these are images of the DNA double helix. You can see the helical repeat here in these little blobs along the polymer axis. And this was published in Science in 1989. Two of the principal players, Tom Tundat here and Larry Nagahara, as photographed as part of the ASU group at the American Physical Society meeting in St. Louis in 1989. There's somebody else, though, I should feature here, is Lynn Oliver, who was a solid state physics postdoc in my group at the time, working on laser spectroscopy. And I was puzzling over the fact that the electrochemistry was incredibly hard, and we seemed to be able to find people who knew physics but no electrochemistry, and electrochemistry but very little surface physics. And so Lin had said to me, well, who's the smartest person you know? And my reply was, well, Nongjain Tao, of course, but he doesn't know any electrochemistry. And Lynn Oliver said, well, give him 6 weeks and he'll know more than you. And Lynn was right, and we'll come to that in just a second. But another aspect of this imaging of DNA that raised a lot of questions was just the magnitude of the current through the molecule. I had a discussion about this with Ivar Gaviar, who in his salty language pointed out the conventional understanding of tunneling at the time, which is that the electronic decay should be fixed by the work function of gold of 5 eV. So the decay length should be about half an angstrom. And if you put in 2 nanometers as the height of DNA, and then assume the quantum of conductance for a point contact, the conductance over a piece of DNA should be 10 to the minus 24 Siemens. In other words, 10 to the minus 12 picoamps for 1 volt applied. Clearly absolute nonsense. I'll come back to this later. But let's return to the electrochemistry and the fact that we needed to learn, and we needed a very smart person. I was lucky enough to hire Nongjian back from a solid state physics postdoc at City College in New York. And Nongjian very wisely chose not to tackle the DNA electrochemistry problem, but to look instead at simpler but very well controlled electrochemistry on surfaces like the gold 111 single crystal surface. And so here is a series of images taken for halide adsorption on the gold 111 surface. And what you see here is this very nice reproducible voltammetry for absorption and stripping of the halide adsorbate on gold, and correspondingly beautifully reversibly one saw these images here of the bare gold and here of the iodide absorption in this case. Nongjian went on to produce a whole series of quite remarkable electrochemistry papers and left my lab in 1992 to move to Florida International University. Now, FIU at the time had no graduate program, so you can imagine how startling it was when this incredible paper here came out in Physical Review Letters in 1996. Notice it has just one author, Nongjain Tao. And what Nongjian did in this work was to show that on graphite, so here's an image of the graphite lattice, that as you increase the concentration of hemine molecules containing iron, here in an adsorbate without iron, you got an increasing density of bright spots, and you saw the cyclic voltammetry change amplitude as you increase the number of active molecules on the surface. In particular, what he found was, if he examined the brightness of these spots here as a function of the potential of the surface, as he passed through this beautiful reversible redox peak here, the STM image became most intense. So he showed there was a resonant tunneling effect through a redox center, a quite remarkable single author work by a junior faculty member, for which Nongjian quite rightly won the Helmut Fischer Medal in 2003. What about this question of the impossible conductance of DNA, though? Well, it wasn't just DNA. David Kay and myself compiled values for single molecule conductance measured by various labs around the world. And here's part of that table in that review, where we list molecules for octane dithiol. And what you see is that the current per molecule at fixed bias covers 5 orders of magnitude, same molecule, but just different labs. And the question was made crystal clear to me when my theory colleague, dear friend and brilliant colleague, Otto Sankey, asked me the very simple question, where do I put the atoms in my calculation? And so it became clear that this was the key issue in the conductance of single molecules. And so I set out in collaboration with the Morgust chemistry group here to conduct the following experiments carried out by my then graduate student, Xiaodong Sui, You'll notice many of you watching this, other familiar faces around this Thanksgiving table at our cabin in 2001. And here's the experiment that Xiaodong did. He made a monolayer of octane thiols on a gold substrate, but included a small concentration of octane dithiols so that when you imaged the top of the film, you could see the protruding sulfur atoms. He then incubated this with gold nanoparticles and used a gold-coated AFM probe so he could form a gold well-defined bond molecule, well-defined bond gold circuit. And when he measured the current voltage curves, he'll find that they clustered in groups such that if you divided any one group by the appropriate integer, you could reduce all these curves to a common curve. And so what he was looking at was the conductance through 1, 2, 3, 4, 5 molecules. Notice this flat part of the curve here. I'll come back to that in a couple of slides. Well, Nongjian, as ever, improved on that work brilliantly by taking the statistical analysis we had developed and applying it to a break junction. The result was a much simpler way to measure single molecule conductance. And this paper here that has been cited now more than 2,000 times is Nongjian's seminal contribution to single molecule electronic measurements. I mentioned to you that we had this flat region of conductance in our current voltage curves, and we found that the edges of the curves agreed with Nongjian's data, but Nongjian did not see this flat region. And so it occurred to me that the gold nanoparticles we were using were pretty small. And if one estimated the size of the band gap in them, it was greater than KT, and therefore these particles should show a Coulomb blockade. Well, Tomo Morita, who was visiting my lab, and here's Tomo spending Christmas with us pulling English Christmas crackers, Tomo synthesized much larger gold particles such that the band gap was smaller than KT, and now the IV curves became completely linear with no flat region, and our data agreed completely with Nongjian's data. Nongjian, meanwhile, was moving on to bigger and better things, and with Piming Zhang from my lab, he devised a way to measure the conductance of small DNA molecules by terminating them with thiols, one on each strand, and then he could see peaks corresponding to 1, 2, 3 DNA molecules in the gap, and hence measure the conductance of a given DNA molecule. Fast forward to 2015, where Nongjian has moved these measurements to a state of elegance. Here you see the resistance in megaohms as a function of the number of repeats. For in these black squares here, a DNA that consists of alternating CGs, and in these blue symbols here, a sequence that consists of NCs followed by NGs. What you see in the case of the black squares is what is expected for hopping transport, which is a linear increase of resistance with distance. In the case of the blue symbols, you see something quite different, an oscillating conductance as a function of length, which with Mark Ratner and Vladimir Mujica, Nongjian interpreted as semi-coherent interference effects in these shorter chains here. Nongjian also, in 2004, turned his attention to peptides. And so here is part of Nongjian's paper on measurements of short peptides, once again thiol terminated. And what he saw was very interesting. These peptides, as a function of length, the current decayed very, very rapidly indeed, slightly more rapidly than even in an alkane chain. And certainly the contact resistance, if you extrapolate this line to 0 length here, was higher than the contact resistance of an alkane chain. So this confirmed what I think probably most chemists in the world believe, and I certainly believed strongly, which was that proteins must be insulators. Certainly peptides are great insulators, but this, as I said, convinced me that proteins were surely insulators, and the reports of conducting proteins must be some kind of artifact. Well, I was shown to be wrong here by Yanan Zhao, a graduate student at the time. I had suggested to Yanan that maybe we could make a biosensor by making a very small electrode gap and functionalizing with a specific ligand for a protein. So we happened to have integrin in the lab, an RGD target peptide that was thiolated that we could functionalize the electrodes with. And so the idea was when a protein bound, maybe we could see some disturbance of the tunnel current through these electrodes. Well, it turned out we saw a lot of disturbance. So here is a trace of a pair of electrodes in phosphate buffer, here in phosphate buffer, but containing a mutant integrin that does not bind RGD peptide. And here now introduced a normal integrin that does bind RGD peptide. And what Yanan saw was that above 100 millivolts, the conductance fluctuated wildly by amounts of many hundreds of picoamps. Now the gap here is about 5 nanometers. We're under potential control. There are no Faradaic currents. And this is quite specific to the binding of the protein. So hundreds of picoamps at a fraction of a volt is a conductance of over a nanosiemens. And we know, because we can take TEM images of these junctions, that the thickness of this gap was about 5 nanometers. A conductance of a nanosiemen over 5 nanometers was just inconceivable. And I didn't let Yanan publish this because I simply did not believe it. Well, Bintian Zhang joined our group and repeated many of these measurements, as well as doing many of the controls that forced me to realize that Yanan was right. So Yanan, I apologize to you. We did finally publish a paper with your name on it here, showing these giant conductance fluctuations in proteins. Now in this work, we had quite a large background current because of the way we made the chips. So in a subsequent paper, we got around this problem. We had a very low leakage system, and we could measure the conductance of single molecules very well. And in this paper, we showed that we got no conductance at all unless we had at least one specific, by which I mean well-known chemical bonding contact to the molecule. We found that all non-redox active proteins we looked at, and we deliberately looked at proteins without redox activity to avoid electrochemical currents, we found they had similar conductances, depending mainly on the contacts. With one specific contact and some second non-specific contact, presumably like a lysine group interacting with the metal surface, we saw a conductance of about 0.2 of a nanosiemen. With two specific contacts, the conductance was about 10 times higher at 2 nanosiemens. Interestingly, it varied very little with protein size, showing that the contact resistance was the dominant contribution and that the decay of current inside the protein was small. And we found also that the conductance did not change very much as we changed the gap size until the gap got so big that the protein became disconnected. This is very unusual in STM, but understood here in the sense that we had two specific contacts. And so the protein just bridged the gap regardless of what it was until the gap broke. We extended these techniques to measure the decay length in this paper here in nanoletters. I just want to show some more up-to-date unpublished data in which we synthesized a series of molecular wires all the way out to 20 nanometers in length and measured the decay of current with length. And what you see here quite clearly is that the conductance decays exponentially. On this log plot, it's a beautiful straight line. This is what you expect for coherent tunneling, which seems to be completely impossible over distances of 20 nanometers. If you look at the resistance versus length, like Nongjian did, unlike DNA, which looked linear, a linear increase of resistance with length, Ohm's law, as expected for incoherent hopping, you do not get a good fit. We don't understand the origin of this, and it's the subject of current work in the lab. One other feature of the conductance of these non-redox active proteins that is remarkable is, lo and behold, we reproduce Nongjian's resonant tunneling curve, but without a redox factor present. We stumbled across this because we found that the signals we got from molecules trapped in the gap were very dependent on the metals in the gap. And so when we changed the combination of metal electrodes and measured the potential of those surfaces by measuring their rest potentials, so that we could then calculate an effective potential for the various pairs of metals, we got this beautiful resonance curve. Now, this is a somewhat difficult experiment both to carry out and understand. So we could reproduce this curve by taking one pair of metals, in this case palladium, and controlling the surface potential with respect to a reference electrode, and we duplicated this curve. So there is a resonance. It's about 0.7 of a volt removed from the nearest redox process, which is the oxidation of tyrosine and tryptophan. And so what we conclude is that we are seeing transport through the unrelaxed states of the molecule. In other words, the Marcus lambda is close to 0. And this may have something to do with the fact that the length dependence appears to be more in the quantum mechanical limit than the incoherent hopping limit. So let's turn finally to applications of this technology of single protein molecule electronics. And I want to show you some work we have been doing with a DNA polymerase. This was published in ACS Nano. So if you're interested in the details of the experiment and the controls, you can find them in this paper. But briefly, what we did was to functionalize a pair of electrodes with biotin. We then captured streptavidin on the electrodes. So now our electrodes are passivated with streptavidin, so our polymerase with its surface cysteines can't interact with a metal. But we made a polymerase with biotins at 2 points on the polymerase separated by about 5 nanometers. So we have a doubly biotinylated polymerase. These streptavidins, of course, combine 4 biotins. And so we completed the junction by having a polymerase bridge the gap. Now, when we made junctions like this, we were able to record fluctuations of the enzyme. So here's a current versus time recording showing those fluctuations. Now, even when the enzyme is not active, there's a background level of fluctuation. This is not random noise. It's 2-level system noise, but it's been steady like this for about 20 minutes. We have the enzyme sitting there with a primed DNA template and magnesium ions in solution, so we're all ready to go. And then we add the complementary nucleotide triphosphate, and we wait for a little bit. And what we'll see is the conductance doubles when the polymerase goes into its active state. Those are those high current features there. Notice in the high current state that the noise is much larger than the noise on the baseline. Let's just let another burst of activity come along. There we go. We believe those features are associated with the nucleotide incorporations of the polymerase. And so we are funded by the National Human Genome Research Initiative to try and develop this technology as a DNA sequencing technique. So I started by showing you the great collaborators of the early days at ASU. I'm well past my sell-by date, but I am privileged to have great collaborators and lab mates. And so I end by showing you this 2021 picture of a lab get-together. This was the post-COVID vaccine picture. So we've got not everybody here because not everybody was vaccinated at this point. But I am still enormously privileged, lucky, and happy to be working at Arizona State with a great group of people. Thank you for listening.